Thursday, we will be meeting in Klein 121, and that's starting at 1:45 p.m. If any of you are not able to meet either in person or on Teams on Thursday at 1:45 p.m., please do let me know that, because I have been assuming that everyone has that time open. But if you don't, just let me know. And I will be recording those sessions in some way so that you can watch later. But I think it's more beneficial to be there in person or on Teams Thursdays at 1:45 if you can, in Klein 121. And what we will be doing this week in lab is some instruction on how do you find the papers that you will need to cite for your project. And my friend Emily Krug from the library is going to be there doing some of the instruction, and I will do some of the instruction. But before Thursday at 1:45, I would like for you to pick your figure. So I've gone in and commented on all of your papers, and you can pick which paper you want to do. But from that paper, you will need to pick your figure and upload the file of the figure to Canvas. So take a screenshot or whatever is your way of cutting out something from a big paper and upload the file to Canvas. If you want to upload like the big picture file, you could, but I'd actually rather have the little cutout part that you're going to use. And also, I will want the figure legend with that. The other thing that I would like for you to have ready by Thursday is what is the author's big question? So this would eventually become the beginning section of your paper when you're talking about. These authors are trying to understand eye development in Drosophila, and it's probably better than that, but something like that. Or these authors are trying to understand better ways to give babies born by C-section the appropriate flora. Emily, I'm not saying you have to pick that, but if you did pick that, some context material, like what is the big question of the paper? And if you have that on Thursday, then you can be focused on, okay, for my figure, what are the things that I now need to look up and learn about the figure? So that's the plan for Thursday. Remember, it's in Klein at 1:45 Thursday. Any questions about your paper stuff so far? I'm excited about your all's range of topics, let's say. They're really interesting. I always learn a lot of new stuff from you all, reading your all's work on these projects. How many slides did I have left in the last? No way. Yay, me. That's great. I'm consistently like 3 slides behind in anatomy and physiology 1 every time. And it's just enough that I can't skip over that, but it gets tedious. Okay. So we're starting a new topic, which is cell signaling, and I'm pretty sure that some of these slides will spill over into Thursday. But cell signaling is one of those key topics that that you learn in cell biology, and what I'm wanting overall in this unit is that you learn in concept how cell signaling works, and you learn what are the major parts of cell signaling networks, so that in the future, when you come to some new kind of cell signaling process that you haven't seen before, you can have some familiarity with the major parts and think, oh yeah, we learned about that protein and how it works. And you get a toolkit to understand any kind of cell signaling that you may need in your future. So the point of cell signaling is to let cells sense what is in their environment. The cells need to know what's going on in the environment to monitor what is the world like outside of me. And they can also receive information from neighbor cells. They can get information specifically just from the environment or also from neighbor cells. And they will take the information and process it and then do something with it. So it's, in a way you can think of it as input. They're getting some kind of sensory input. And then they process the info. And then they develop some type of output. And the output, a lot of times, is going to be changing their behavior in a way that reflects the information that they got. And all cell signaling would follow that kind of pathway. In this class, we focus more on this part. This is still important, but the lectures are going to be focused more on the input and the processing. And when these signals are coming from outside the cell to the inside, the membrane in a lot of cases is a type of barrier to the signal. Not always, but usually. So the difficulty of getting the signal across the membrane and into a form that the inside of the cell can perceive it, that's called signal transduction. So we could say that this part here could also be known as signal transduction, getting the information into the cell.
But it says and to get into the final target cells, so wouldn't that be the output?
Final target molecules. The target molecules are usually the point where you kind of make the turn to turning the information into the output. Yeah, so what the target molecules are usually, we're going to see these pathways and they're oriented vertically. They're not really, but in books they are. And then down at the bottom is the target, and then the target will then enact the output. So the targets can be all different, the signals can be all different, but it's the in-between steps. The cells use kind of a smallish set of signal transduction proteins to relay these kinds of info. So when cells are communicating, there are four big styles of cell communication, or four big styles of signal transduction. And the first two, well, really the first three, have to do with floating signals or diffusible signals or soluble. So when you see in here red dots drifting from place to place, those are diffusible signals. They're kind of coming from one place and they float through a liquid. It might be long distance or it might be short distance, but those are diffusible signals. This one is different because the signal is not floating around the signal is attached to another cell. And this one requires the two cells to basically touch and pass information through contact. So that one's called contact dependent. All of these others, the two cells that are sending the signal and receiving it, do not have to be touching. So I want to say some more information about these three styles. If the signal passes through the bloodstream on the way to the target cell, and it's small and diffusible, that's hormone signaling. And we call that endocrine. Endocrine is a hormone, which is a small diffusible signal, and it has to travel through the blood before it gets to the target cell. So this is long distance. If the signal travels a short distance from the, what's this called? The sending cell to the receiving cell, got it backward. The signaling cell to the target cell. You see, if the signal's still floating, it's not going through the blood, it's going a short distance and it's local. That's called paracrine. Paracrine. And if the short distance floating signal is coming out of a neuron, it's really paracrine, but it gets its own name, it's neuronal signaling. So that case, everything neuron and brain gets its own names. The signal is a neurotransmitter and it's coming out of a neuron, but it's not any different than paracrine actually. These seem so different, but they're all keyed in on this event, which is the signal, which is shown in red in your book, binding to a receptor to receive that information. So you could circle here, circle here, circle here, circle here. The nature of the signal can be different, but the initiating event in signal transduction is similar in concept for all of those four types. For hormone signaling, there are so many kinds of hormones. There are many, many kinds of hormones. They're coming from different locations of the organs that make them. They have different chemical structure, and they do different jobs depending on where they are received. But what you should know about hormone signaling just out of this class is that they float, they have to go through the bloodstream, and it's usually not the same place that makes the hormone and acts on the hormone. So for example, insulin is a hormone and the pancreas makes it, but it's cells all over the body that respond to it. But the signal, whatever it is, it has to go through the bloodstream. You'll learn more about specific hormones in mammalian physiology if you take that, or even plant biology has hormones. The point of this chart is not for you to know details of the chart. It's the top words, and this is just examples of certain hormones. This particular chart is listing some of the other kinds of signals and whether they are local. Whether they are local paracrine type signals or neurotransmitter signals or contact dependent. This is kind of a list of the most famous and the most well-studied other types of signals that aren't hormones. And again, the point of the chart is not for you to know the details, but these are kind of the best studied other kinds of signals besides hormones. So we see on here, we're looking at a particular chemical, acetylcholine, which is the red dot. And your book uses green to depict the receptors for a certain signal. And notice here that there are different kinds of receptors. So this is a certain kind, that's a similar kind, this is a totally different kind. All three of these can bind to acetylcholine. But when they do bind to acetylcholine, they are going to result down the line in a different action or a different output of this target cell. In your heart, acetylcholine slows down the electrical signals. In your salivary glands, acetylcholine makes you produce more saliva. In your skeletal muscle cells, acetylcholine causes them to contract. So what's different in these is the receptors can be different, and what the receptors do with that information to process it then is producing a different output. And this type of variety comes from the cells have some mix and match signaling components. So they can have different receptors, different styles of receptors, different styles of intermediate signalers, we will say. Different styles of integrating processors. What I'm trying to say by this is that we don't need a separate new individual unique signaling pathway for every single thing that our body does. We can put together a signaling pathway that has a certain type of receptor and a set of 4 intermediate signalers and a couple of different integrating processors. And the variety of the combinations of those give us multiple outputs. So this set together can receive multiple inputs and enact multiple outputs. And that, when scientists basically finished the human genome sequencing back in 2000, like sometime around year 2000, they were initially shocked that we're not complicated enough and we don't have enough kinds of genes to do all the cool things that we do. This kind of thing where we use a few parts put together in combinatorial ways to give multiple outputs. We don't have to have as many genes as you might think to be an awesome human. So for any given signal transduction pathway, there is a signal and there is a set of parts that allow that to happen. But if you learn the basics of the parts, you can understand any signaling transduction pathway. Another way that cells use signals is that they are able to interpret multiple signals and process them, processes underneath. They can process multiple signals at once to produce different outcomes. This picture is showing letters that indicate the signals. So every letter is a chemical. And it's saying that if a cell gets the combination of signals A, B, and C, the sum of those, or the outcome of that, is that it just stays alive. It just maintains what it's doing by a combo of A, B, and C. If the cell gets the maintaining signals plus D and E, it will stay alive and grow bigger and divide by that particular combo of signals. If the cell gets A, B, and C to stay alive and F and G, it can turn into a different kind of cell or differentiate. This would be a good word to define. Differentiate means it turns into a different cell type. Differentiates. And if a cell gets no signals at all, if it is basically cut off from outside information and doesn't get signals, that is dangerous, and the cell is supposed to die. So without any signals and any outside input, the cell is supposed to die, because that's not the normal situation. A lot of cells in our body that could be cancerous don't make it to be cancerous, because the default is, if you're not doing your normal signal processing and receiving information from the outside, you're supposed to undergo apoptosis. So the cells, they're acting like almost like a computer network. Like, if A, B, and C, then survive. If A, B, and C, and D, E survive and grow and divide. So they're integrating multiple signals at once to have a certain outcome. When I don't have quizzes and I suspect that you haven't read, I will usually try to let you take in the figure at first. So this is starting to flesh out how we look at cell transduction, signal transduction. We've got the red dot signal. We've got a receptor that detects it and binds to it through the four forces. And then when we have arrows coming off of that initial event, these are representing the signal transduction pathway, or the components. This stuff that I've called intermediate signalers. And later on, we'll look at what are some of those players, but for right now, they're just depicted by multiple arrows. Like, there's this, and there's this, and there's this. That's the signaling pathway. But you can divide cell responses to signals into two major styles. So either it will be the fast style, where the signal is changing a protein function that is already there. And they're calling this altered protein function. That's specifically of a protein that's already present in the cell. Do you ever learn about those like Pavlovian dog experiments in psychology? That little sound is, like, if you play it for me when I'm 80, I'd probably like, what am I supposed to do right now? I need help. Okay. Oh, this is line up now. Yesterday I had a class, and somebody had joined it on Teams, and they were having this loud conversation and all this sound, and I said, so-and-so, and they didn't say anything. And then I just keep teaching, there's all this weird sound. So-and-so, can you mute? And they still weren't listening, so I just turned them off. I felt kind of messy about it. But, let's see if somebody is watching. This would actually be a good way to troll me, I guess. I like it. I don't see anything. Okay. There we go. So this style, whatever the output is, can be fast because this protein that's being changed is already in the cell. So it could be that there was a protein that was already there and then it got phosphorylated and that turned it on. Or there was a protein already in the cell and the outcome of this is that it got a ubiquitin on it and then it goes to the protease, the trash, so to speak, and that changes what the cell is going to do. So if that's the type of response and the protein that's the real target is already existing, the cell can respond to that signal really quickly. This does not require new translation. This doesn't require gene expression or translation. And one of you turned in one of your two papers, and they were using experiments to determine whether the process depended on new translation. And now I don't remember who that was, or what paper it was, but I'm hearing a lot of like Teams bloops. If you have a question or something, just unmute and say it. Because it may be glitching today or something. So that's the fast style. The slow style requires that the signal change something about transcription of genes and that the signal eventually be transmitted into the nucleus. So if the cell is gonna need to transcribe new genes or change the levels of transcription and change protein synthesis, that's another way that cells can respond, and that's a slow response. This is also more permanent. When you're 14 and you have a lot of different hormones in your body and you like yell at your parents and you hate them, that's like a long-term slow change in your brain autonomy and your own person and stuff like that. That's a slow change. When we sense that we're getting extra glucose in the blood and we need to release insulin from a cell that was already there, that's a fast response. So hormonal changes tend to be more of the slow response, but some of those other signals can create a fast response. It just depends. Does it require new proteins? Does it require gene expression? Either style is going to change what the cell does and give it a new behavior or a new response. So no matter what the signal is, most signals in the world are not able to cross through the membrane on their own. So we learned in the last unit that the plasma membrane blocks most substances from just crossing on through unless they're small and hydrophobic. So there are a few signals that can cross straight through a membrane, but most of them are going to be staying on the outside of the cell and bound by a cell surface receptor. And these two would bind together through the four forces. It's non-covalent. It's usually a temporary binding. But the binding event is going to change, in some way, change the shape or change the function of this receptor protein, and that's how the signal gets across. So in essence, you have binding changes the shape slightly of this receptor, and that's going to change the signal that gets to the inside of the cell. So binding out here leads to a change in here. That's how transmembrane receptors work. And the binding region has to be on the outside of the cell where the signal molecule is.
So the signal doesn't actually go inside?
Most of the time, the signal does not go inside. Yeah. And that's a pretty big concept, that most of the time the signal is coming from the outside and it actually stays on the outside. But the binding event to this transmembrane protein carries the signal information to the inside of the cell, even though it doesn't literally go in. But once in a while, certain cases, if the signal is something very small and hydrophobic, and it can go straight through the membrane and diffuse everywhere, those signals tend to have a receptor that is inside the nucleus. This is kind of more rare. This is not the usual, but there are some signals that can diffuse all the way into the nucleus and bind to a nuclear receptor and start the signal transduction from that point. But they've got to be really small and hydrophobic. So nitric oxide, NO, nitric oxide acts like this. It can get straight into the cytoplasm, but that's a little bit more rare, to not have to require the transmembrane receptor. Some other examples besides nitric oxide that can go through the membrane are steroid hormones. These are steroid hormones. They are fatty. They are basically small, and they're able to go straight through membranes. These three are derived from cholesterol. Thyroxine is not. But these would be some of the few examples that don't need an extracellular, they don't need an extracellular receptor. This one's part of thyroid hormone, thyroxine. Picture of cholesterol. You know, sometimes it makes that noise because, it makes that noise because it gets slow. That was somebody that got kicked out and had to come back in. I don't have anything particular to say about cholesterol, just that shape contributes to making these hormones. So for those steroid hormones, like cortisol, they do not have to go, or they are able to go straight through the plasma membrane, and they come to the cytoplasm, and they bind to a protein that's kind of waiting out here for them, that's called a nuclear receptor protein. A nuclear receptor protein. And the combo of these two is allowed entry. The combo of those two is able to go through the nuclear pore complex. So at this point, these two together could bind the nuclear transport receptor, go through the nuclear pore complex, and the combo of those two together is going to act like a transcription factor. That's why they're showing it here. It's landing on the DNA, it's landing on the promoter, and it's going to change gene expression. The cortisol is not a transcription factor. The cortisol is a hormone that can drift into the cytoplasm, and it binds together with its receptor, and that shape combo is allowed entry into the nucleus, where it will then work as a transcription factor and turn on this gene. And for cortisol, these kinds of genes have to do with how we process nutrients. And so it's sort of changing our blood chemistry for how we process nutrients. It is in some ways a stress hormone. So we're going to transcribe some genes, and then the eventual outcome is the big overall response is that we are going to later put more sugar and fat in the blood. We're going to put more sugar and fat in the blood, is what happens down the line. That's the eventual outcome. If you're exposed to cortisol, which is a stress hormone, long term, you will have more sugar and fat in your blood long term, which can make you more prone to strokes, more prone to diabetes, and so on. The hormone signaling tends to have longer term effects than your short term paracrine or neuronal signaling, and so on. Okay. One of your other signals that can diffuse straight into a cell is nitric oxide. And this particular one, nitric oxide, is not a hormone. But what it specifically does is it diffuses into muscle cells, and it causes them to relax. And when those muscles are in blood vessels, the relaxation is called vasodilation. Vasodilation. So the muscles in the lining of the blood vessel relax, it opens up, and you would have a blood pressure drop from the nitric oxide. This process is involved in erection in men, like getting blood vessels to relax, blood to flow into the chambers in the penis that cause enlargement. That's a short-term signal coming from nitric oxide, and it diffuses into the target cells, which are actually in gray right here as the muscle. I'm not going to go into too much more details of that, but that's an example of there is a signal, there is a response, and in this case, it's a short-term response, and it's a quick response. It doesn't require new transcription. So if a cell is going to receive a signal and respond to it and change its behavior. Get it, process it, change your behavior. It's really important to respond in real time. It's really important to sense and respond to the signals in real time. And what I mean by that is the conditions can change at any given moment. So it's really important for the cell not to be stuck in what happened in the signal that it got 2 hours ago, because something might be different now. So any of these pathways, get the signal, process it, change the behavior. It's really important that it be able to reset. So the cell must be able to reset the signal machinery to get new info. Or let's say updated information about what's going on. For example, this is a silly example. If the fire alarm goes off in Liston, there may or may not be a fire, there's probably not. But if no one ever goes and resets them so that they can go off the next time, that's a danger to the building. That's a danger to the cell. So the cells have to be able to reset their signaling machinery. And I tend to call this turning the signal off. And I'd like for you to know, for every major signal pathway that we talk about in this class, yeah, you need to know what turns it on, but you also need to know the off switch, or the mechanism by which it resets. So the example here, we have two different signals going on. We have some acetylcholine, which is a neurotransmitter, and it activates eventually nitric oxide. Nitric oxide will diffuse out of this cell, it goes into this cell, and it quickly causes this cell to relax smooth muscle. So we've got two different pathways going here, starting with acetylcholine. This pathway will turn itself off because nitric oxide in water will not last long. It turns into nitrates and nitrites, and the signal breaks down. So the off switch in this case could be that nitric oxide breaks down. Or it could also be turned off if acetylcholine stops binding over there. It could be turned off here, it could be turned off by the breakdown of nitric oxide. But every signal pathway has to have a way to turn back off so that it can get updated information. This is the best place to have a break right here before we get to this slide. Emily, how long were you off?
For about 5 minutes.
I was hearing a bunch of sounds, and sometimes it makes the sounds if the network is slow or something.
Yeah, my Wi-Fi cut off and then I couldn't get back into the meeting.
But it wasn't too long. And those of you on who are on Teams, I think that the best thing is if you've got a friend in class that has the notes at the correct place on the slides, and they could send you pictures of them or get them in some other way that is most helpful. So. Review what's coming up here. See if my coffee's still hot.
Does insulin turn on cortisol?
Does insulin turn on cortisol?
Off.
Off.
They're opposite, right? Insulin makes blood sugar go to muscles and then
Insulin and cortisol interact with each other, but I don't have the details of it memorized. It could be that cortisol, I think cortisol has some downstream regulatory effects on insulin that eventually can lead to type 2 diabetes, but I don't know the details
I
of
just
it.
know that if one's high, the other one's low.
That makes sense.
I didn't know.
Yeah, I mean, that makes sense
one is
because
secreted
insulin puts glucose into the blood. Sorry, insulin takes glucose out of the blood and cortisol puts it in. Yeah, but there's also, that we know that long-term cortisol elevation also can promote type 2 diabetes. Yeah. Did you learn that in A&Ps?
But the insulin.
Yeah.
Yeah,
Okay.
that and exercise physiology.
Oh, good.
And I think I already knew that before I took the A&Ps too, but not to the extent I know now. But
Yeah.
I knew of it.
There's so many times that I would like I would like to go to Mammalian Phys and just be in the lecture because I never had it myself, or go to Exercise Phys and see what they teach and make sure that my A&Ps get people ready for it. Or go to, the one I really want to go to is the nursing pathophysiology, just because I think it's cool, but that is the biggest driver of how I teach AP2 is for nursing pathophys.
That should be the hardest one of all.
It is for that first semester. But I have too much to do. I can't.
Rutgers, when he taught Vermilion, I don't know if he was part of it, but he did a really good job with explaining.
Yeah. We've been making these pre-lab videos for the A&P labs where they have to be shorter and people then have to come in and be faster about getting their lab stuff done. And so I film the ones for, like, I run the camera when he does the A&P1 and he'll run the camera when I do A&P2. And I always like to see different ways that the other teachers explain stuff. It's nice. Finish this up. Oh my gosh, that could have been so bad. So bad. I almost fell over my purse. For those of you. I haven't fallen down at King in about 2 years now. I used to fall a lot in the dining hall when they mopped the floor. No, I did. Like once a year, they would mop the floor or somebody spilled, and I have certain shoes that are leather soled, and I tend to walk fast. And like those one shoes, I would hit that and just like drop all my food. It's so embarrassing. And I don't wear those shoes much anymore, but this bump thing is always tripping me up, literally. You know, they have those yellow signs that are like, beware, don't fall. Like I might as well put my face on the one in the dining hall. Do not fall. Okay. So this is an overview figure to kind of show how signal transduction pathways tend to be represented. So these signal transduction pathways, you can see them on posters, you can see them on review articles in this book, all different classes, but they tend to look kind of like this. So they start with the signal up at the top. The signal does not come from the top. They do not fall down by gravity, but that's the way that they're depicted. And it binds to a receptor, and that depends on the 4 forces. And the receptor tends to cross the plasma membrane, like so. And the receptor will have a particular protein inside the cell that it sends information to, which is green square. And then green square is sending information to green triangle. Green triangle sends information to green circle. These dark green group are the intracellular signaling molecules, or what I have called over here the intermediate signalers. So the intermediate signalers tend to have some organization that squares tend to send info to triangles, and triangles tend to send info to circles. And there's a certain set of those that are the intracellular group. And then they will either through fast or slow mechanism. We could write in here fast or slow response. They're going to change a behavior. And the proteins that change the cell behavior are called effectors. That's kind of like what I've called over here the integrating processors. They're the ones that are going to change what the cell does. And the major types of effector proteins can be transcription factors. And these would be leading to the slow kind of response. Or sometimes the effector protein is cytoskeleton. And if you change what the cytoskeleton does, the cell can move or the cell can change its shape. So that's one of the major outcomes. Or sometimes the effector is an enzyme, a pre-existing protein. So these would be proteins that already exist. And the enzyme is going to change to be faster or slower, to work better or to work worse. And that is going to change some kind of cell chemical reaction that the cell is doing. So these are the enactors. These are the behaviors that the cell is changing. But what we're going to spend most of our time on are the parts represented by that dark green, the intracellular signaling molecules or the intermediate signalers. And there are less than 10 major kinds of intermediate or intracellular signaling pathways, and we're going to learn about 3 of them in this class, the 3 biggest ones, the 3 biggest networks of intracellular signaling. So here we see another schematic of the general cell signaling process, and it's got a few more details on it. This figure is intended to show what are the variations, what are the styles of how the intracellular signaling works. And so sometimes, up here, we'll start at the top. There's a certain set of proteins that always work together. Up here at the top, the light green. So we've got the receptor, and then there is like an in-between protein. And the in-between protein signals this next one. And this one, see how one signal came in, what is that? 7 signals came out. Or one event was detected, and then suddenly there is this cloud of signal coming out. This kind of step is amplification. And normally amplification will work by this activated protein acting like an enzyme and suddenly cranking out a lot more signal that then diffuses through the inside of the cell. These, in the light blue dots, these are often called second messengers. second messenger. And they can diffuse through the whole inside of the cell and be a very effective amplification. So this would be like if we were playing the old, like, youth camp game of telephone or whatever. You tell your neighbor, I don't know, you tell them something and then they tell the next person and they tell the next person and then somebody screams it out. That doesn't ever happen, but that's what amplification is. And when you see divergence or spreading out of the signal, that's usually an amplification step. Back up to that protein up at the top that says scaffold. Scaffold. So we see that there is this rectangular, they're not really rectangular, but there's this protein that is kind of keeping these three together in a group. It's kind of organizing them in the same location so that they can work better together. That would be an example of a scaffold protein. Holding some together that are supposed to work together and keeping them in the same physical location. That can be pretty common in transduction pathways. Here we have another example of, in some way, amplifying, or this protein is acting on 3 different targets. It's distributing the signal. But the last part I want to mention is right here with guitar pick, light green guitar pick protein. It is sensing 3 signals at once. It is able to be modified by multiple signals, and I want to write about that. So I'm writing on green guitar pick that is receiving 3 signals at once. This is an integrator. Integrator. It is modifiable by multiple signals. It can be modified in a variety of ways. And depending on the combo of modifications that it gets, we could say that its combo of modifications is going to control its behavior. You have a picture, if you've got all the slides ever from this whole class, go back to the ones in the, I'll just go there too, because this is connecting back to something that we saw a long time ago, back in the proteins section. Where's that? Unit 1. RNA to protein. Should be way at the end. No, it's not even in this one. Let's try the previous one. Previous slide set. It's a picture of P53 protein. And I think it's in the proteins chapter. There we go. Here it is, right here. Okay. So this is back in your, if you got your slides from proteins chapter. This is an example of a protein, P53, that is multiply modifiable. So it can get phosphate groups at any of those positions shown by the yellow dot. It can be acetylated in a variety of positions at the C-terminus. It can have ubiquitins for destruction added toward the C-terminus. And it can have any specific different combo of these. So if it had, let's say, 4 phosphates and 2 acetyl groups in a certain position, that changes what P53 will do. And this particular protein controls cell division. It controls whether a cell will divide or not. And it is a central player in cancer networks. So, for example, if P53, had one particular combo of signals, it might bind to others, or it might be able to move into the nucleus, or it might get destroyed by the proteasome. P53, I don't think ever goes to the membrane, but it could do these other things. So that particular protein is an example of the most famous integrator protein in cell biology, would be P53. And it is at the center of cell division signaling networks. And it helps, when it's acting normally, it will help prevent cancer. If it is acting abnormally, that can promote a higher risk of cancer. So a protein like this, the most famous example of the integrator is P53. And we'll learn more about P53 in November when we get to that unit. A lot of the different cell signaling networks involve protein kinases. One of the common ways of transmitting a signal is for a protein kinase to be activated, and it adds a phosphate onto the next player in the intermediate signaling chain, and the phosphate might be activating that one to send the signal onward. So in this case, phosphorylation turns on the next partner in the signaling chain, and the signal is sent on. This can be using ATP, or this can be using GTP. In this case, it's a little bit different. The on signal is when GTP is bound to the protein. GTP at some point will hydrolyze itself and kick out its phosphate and turn into GDP, and that's the off state. So having a phosphate bound or having a GTP bound can be an on signal for the protein. And as long as that is bound, whether it's phosphate or GTP, the protein will stay on, but it can be turned off as well. So if a kinase puts the phosphate on, a separate protein enzyme, phosphatase, can take it back off and turn off the signal. This style requires 2 different proteins. Oh, a little bit higher. This style on the left, no, it's too tall. There we go. this style requires two separate proteins. This style requires one protein that can self-hydrolyze GTP to GDP. And this one we're going to look at in some detail today, an example of this style. There are two different proteins that modify this. One adds a phosphate, one takes the phosphate back off. This style is in some ways more efficient, that this green protein can be bound to GTP, turning it on, or it can hydrolyze the GTP and kick out phosphate and be bound to GDP and be in the off state. This requires a lot less complexity because it's one protein basically turning itself on and off by binding GTP or then hydrolyzing it. Uh-oh, well, we already had a break. So now let's look at some details of specific types of cell signaling pathways. Some cell signaling pathways start when the signal opens an ion channel. So in this case, red dot is the signal. Green proteins are the receptor. And when the receptor binds the signal, that opens an ion channel. We would call that ion channel coupled receptors. And then say that this was sodium. It's more outside than inside. The sodium rushes in, and it would then change the cell conditions on the inside, and that's the start of the signaling pathway. That style is ion channel coupled. And the best examples of those are muscle contraction, and we'll look at that later in this unit. The second big style, which we spend a lot of time on in this unit, is a style called G protein coupled receptors. GPCR. G protein coupled receptors. And this is an overview schematic of generally how do G protein coupled receptors act. They have a certain structure that is transmembrane, which we'll look at in more detail in just a minute. The signal that starts this process is going to bind extracellularly, and you see it there as the red dot. When the signal binds to this G protein coupled receptor, that's the green, the GPCR, it then associates with what we call a G protein. G protein is this yellow-orange blob here. And the binding of these two together activates the G protein, which is shown by the pink burst, active. And when the G protein is active, it then associates with the next partner in the signaling chain. So G protein signaling is one of the most used and the most prevalent kinds of ways that cells send signals outside to inside. And people use these a lot, really a lot. So I have you all learn a lot of details about this because it is so commonly used. So we'll look at some details of that coming up next. But before that, the third major type is enzyme coupled. And I think I want to skip over this right now. I'll come back to this slide when we talk about enzyme coupled. I want to spend enough time on G protein signaling. So we're skipping for right now this slide and this slide about toxins and poisons and starting on G protein coupled receptors. Okay, so we have the green snaky cartoon up there, and then we have a real ribbon diagram of a real G protein coupled receptor. And the orange that you see here, that's the ligand, or that's the signal that binds to this receptor. And humans we have many versions of GPCRs. But they all work in the same way. So our GPCR family of proteins, we have several of them, but they all work in the same way. All of them have 7 transmembrane alpha helices. So if you count the snake up there, how many times it passes through the membrane? It's 7. And each time it passes through the membrane, there is an alpha helix that would be largely nonpolar in the membrane region. But then it has some intracellular domains. The intracellular domains are going to change shape when the signal binds. And they change shape in such a way that they bind to a G protein. We'll look at how that works in a minute. They also have this extracellular domain up here. And the extracellular domain is specific shaped for a specific signal. So up here is extracellular domain, and an individual G protein would have an individual shape in that region that corresponds with what its signal molecule is. So the transmembrane segments for these are kind of the same in the family, but they would have slightly different shapes interior, slightly different shapes exterior that let them bind the signal and then transmit that information. But your book is going to use the sevenfold snake depicts a GPCR. And even other figures from other books, they tend to have that 7. So when you see it, it's probably a GPCR.
Does it start when it starts building?
Let me get it now, Mark and Rachel. Yeah. Okay, so there's 1, 2, 3, 4, 5, 6, 7. Yeah. And then the in-between regions form the intracellular and the extracellular domain. Yeah, I usually do trace and count it. I thought, it's too high. It's too high. This picture, this should not really be on this page. I just couldn't get it cut off. So block that out or put the parentheses. That's going with a separate step later. The signal is not intracellular. It's misleading. So for a particular G protein, it's kind of waiting in the membrane for its signal to come. And somewhere nearby, it is not actually this far away. It would be a little bit closer. Somewhere nearby, there are some partner proteins called G protein complex. G protein complex. And this is being shown on this figure as being called inactive G protein. G proteins are always supposed to be near to a GPCR, and they have some particular subunits to them. They have some particular subunits, and we have multiple of these as well. But they work kind of the same. So all of this together is a G protein complex, and it has certain subunits. So this one is called alpha. That's the alpha subunit. And there is also always a beta and a gamma subunit. The alpha and the gamma are held into the inner leaflet of the cell membrane by having a lipid link to them. The beta subunit is held in with these two only by the four forces. So this is a quaternary structure that together form this G protein complex. And the alpha subunit, alpha can bind GDP, which is kind of like an ADP, but with a guanine base. Or GTP. It can exist in two states. When alpha is bound to GDP, this is the overall configuration of the G protein complex. And most of the time, a G protein complex is going to be in this configuration. This is the waiting, like waiting for something to happen, or the off configuration, or inactive, or closed. So maybe let's write all of those. This can be referred to as inactive, closed, waiting, that's my terminology, nobody else says that, or off, when it's bound to GDP. And this cartoon is showing that the G protein is off and waiting, and the G protein, GPCR, is also inactive, waiting, off. The signal has not come yet. This is just the in-between time, that configuration. So when the signal comes and arrives at the extracellular side, it binds to the GPCR and it GPCR shape in such a way that several different things are going to happen. The signal binds, the GPCR changes shape so that it now is the G protein complex. And so that's why suddenly you see that the GPCR and the G protein complex are now together. That's because the signal binding changed that intracellular region. This binding changed this intracellular region's shape in such a way that now the G protein complex associates or binds with the GPCR. And when it does that, it changes shape in the G alpha region to eject or dissociate or to let leave that GDP that it had been bound to. So GDP leaves, and that gap is then filled by a GTP. This is coming out of solution. So the GTP had been floating in the cytosol. And when G alpha is free, open, it has a really strong binding association for GTP, and it will bind GTP that has been floating in the solution. When GTP binds, that causes a lot of drama, a lot of things to happen. GTP binds, and it causes alpha to come apart from the beta gamma. GTP binding is the key event that causes this G protein complex to be open. So we can circle this configuration. This is open, this is active, this is on, not waiting, it's on. So it's open, it's active, it's on. And when it's open, active, or on, alpha by itself or beta gamma together can then send the signal to the next intermediate signaler down the line. Sometimes it's alpha, sometimes it's beta gamma. It depends on what kind of signal came in here and what else is in the cell.
So what kind of pathways usually use GPCR?
GPCR, like what kind of pathways? GPCRs tend to act with certain other workers, let's say. But what comes out of this, like what the outcomes are really diverse. So acetylcholine signaling can work this way. I don't know the other examples right now, but it's really a lot of things. So it's not like GPCRs are always used for neurotransmitter detection. They can be used for hormones, they can be used for all different things. So it's too diverse to say this is the usual style for this kind of thing. The video, that animation video with the annoying music, getting a white blood cell to slow down and squeeze through the capillary wall uses a GPCR signaling. So it's really diverse into like what kind of things depend on GPCR signaling. But that's a good question.
Which part did you say came from just solution? Was it the G?
Yeah, the GTP. Yeah, so this machinery is basically stuck at the membrane. So far what we've got as soluble parts are the signal is usually soluble and the GTP is soluble. But everything else so far, it's either literally stuck in the membrane or it's associated and it's kind of waiting there for the signal to come. As soon as this happens, as soon as GTP binds, GTP binding starts a countdown until GTP is hydrolyzed. So as soon as the GTP floats over from solution and binds to the G alpha, it's on the clock. Like it will be there for a certain amount of milliseconds or something until it's hydrolyzed to GDP and a phosphate. So this, I don't know if y'all like Mission Impossible movies, like Han and the boys, that's one of their favorite things to watch. They just love those. And the old thing with Mission Impossible movies is you get the instructions, like this is what you're supposed to do. It will self-destruct in so-and-so, and it blows up or whatever. GTP binding is like that. Like that's the signal, and it will persist for a certain amount of time, but then it turns itself off by hydrolysis. And that's what we'll look at next. There's just a lot on this slide. Okay. So this slide, we are no longer seeing the GPCR or the starter signal, but they have not disappeared. They are still there. It's just that your book is not showing them right now so that you can focus on this next part, but they're still there, and they would still be very nearby. But once alpha is bound to GTP, remember, G protein complex is going to come apart. It's going to open up. And it's actually a swinging motion. So it's really like, it's not that they drift apart, they kind of swing apart from each other. And while GTP is bound, alpha, in this case, is associating with this new protein that looks like, I don't know, like a brown fishhook or something like that. This is the next signaling partner. And we'll talk about what this is in just a minute, but as long as GTP is bound to alpha, it's going to associate with this partner and be sending the signal onward until GTP is hydrolyzed. And that's an automatic process. So it binds, there's a certain amount of time that it's going to stay in that configuration. And in that short little amount of time, G alpha can associate with the next partner and send the signal onward. But eventually, that GTP will hydrolyze into GDP, which stays bound. The phosphate that comes off floats away. So this is immediately soluble and it floats away. And as soon as GDP is the bound version, alpha gets back together with beta gamma and they form the closed configuration. They become inactive, closed, off, and they go back to waiting. Likewise, brown fishhook, as soon as it's not bound to G alpha, is also off, waiting, inactive.
This might be kind of a meaningless question, but is there ever like a problem that happens with the G protein that it dissociates so quickly that it never reaches the target protein?
That GTP binds too fast, or sorry, that it hydrolyzes too fast?
That it never reaches it.
There are a lot of different things that can go wrong with that binding event. And I don't know of some specifically, but that's definitely possible. So if a G alpha subunit had a mutation where it doesn't, it does not do a timely turnover, let's say. If it's not doing a timely turnover of GTP, that can interfere with signaling. Whether that's survivable by the cell depends on how bad is the mutation and how bad does it mess things up. So if it was really bad, like it never bound to GTP or it immediately hydrolyzed, that's probably not survivable. And we wouldn't see those in animals or people because it's deadly, if that makes sense. But there are a lot of things that can go slightly off. If it binds and it's supposed to last 40 milliseconds and then it only lasts 20, that's probably okay, but the cell is not going to operate properly. We will pick back up with this next time to talk about what happens next and what these players might be. But all G protein, all GPCR pathways start out this same way. That part is always the same. I'll see you on Thursday morning.